Everybody, this is the third and final video for aerobic respiration. So we're going to talk about the third step, which is oxidative phosphorylation. And so we are now down here on the bottom, okay? So it's going to be called oxidative phosphorylation. And you're actually going to need quite a bit of this space. We're about to draw something. So here's what I'm going to recommend. I'm going to turn off this video in a second and start drawing on the PowerPoint, and I'm going to explain to you what I'm drawing. But I suggest you put your pen down and just watch. So you can see how big you're going to need to draw this, and so you can listen. This is normally the hardest part for people to conceptualize because there's just going to be a lot going on in oxidative phosphorylation. So I normally suggest everybody just listen as I run through it the first time, and then I'll be like, okay, pause the video, draw this picture, play it back, and I'll walk through it again another 1 or 2 times, so that way you can then be drawing along and stuff. But this is probably the most abstract part of this process, so it is good to just kind of sit down and absorb what I'm saying. But again, we are on this last part right here, this pink part on my paper, okay? All right, so let's see, video off. Let's go ahead. Oops, I forgot to turn the transcript on. So all I was saying, and for those of you that's using the captions, I was just saying we're starting the last step, and we're using the last part of the foldable. Okay. Here we go, let me turn off that. Okay, so for oxidative phosphorylation, there are 2 big parts, okay? So oxidative phosphorylation, I'm going to write this kind of small, it's going to be the electron transport chain plus a process known as chemiosmosis. Okay, so what we're going to draw is a membrane. I'm going to need to make sure I leave myself enough room for all this. All right, and so in this membrane are some proteins and other molecules. So we typically call them number 1, number 2. Got this other molecule up here you don't need to know the name of. And then we're going to have molecule 3. And molecule 4 looks like a tooth, so I always felt obligated to draw it looking like one. Okay. And then we're going to draw something else on the end here. I'll go ahead and draw it now. It has an interesting shape. Okay, I'm not going to worry about the shape. It's okay. And this is going to be ATP synthase. So notice the last 3 letters of this name, ASE, so it's an enzyme, and it is the enzyme that makes ATP. Because I've been telling you guys that respiration is how organisms mass produce ATP. But so far, you've only seen 2 molecules of ATP made during step 1 and 2 molecules made during step 2, right? And that's not a whole lot. That's not mass producing, right? So it's actually in this third and final step that we're going to make a lot of it. Okay. So let's talk about what's been going on so far. So in the previous 3 steps, we talked about stored energy in the form of NADH and FADH2. And so I told you all they were like little taxi cabs, and they were going to be dropping off those electrons somewhere. And this is where, okay, at the electron transport chain. Okay, and so this is happening for eukaryotes in that cristae, the inner mitochondrial membrane, those cristae, or for prokaryotes, it's happening in the plasma membrane. Okay, and so that's what like these top and bottom lines here, that's showing you the membrane. Okay, but I wasn't going to draw all the phospholipids because that would just be distracting. And so then these proteins 1, 2, 3, and 4 are in 3 of them, right, are embedded in the membrane. They are transmembrane proteins. And then we have little peripheral protein down here, number 2. Okay, and so what we're going to have, what I'm about to kind of sketch, is a bunch of redox reactions. Okay, so remember, oil rig oxidation is when a molecule is losing those electrons. Reduction is when they are gaining. Okay, so let's talk about our two little taxicabs, NADH and FADH2. So NADH is going to drop off its 2 electrons here at protein complex 1. So once it's dropped them off, right, it becomes empty again, and it's going to be NAD+, and it's going to hightail it back out to other parts of the cell to keep collecting electrons as the cell is continuously breaking down glucose and other molecules. So when NADH loses its electrons, right, NADH has become oxidized. It's given up its electrons, but it's given them to protein complex 1. So protein complex 1 is now reduced because it's gained them. And now there's like a game of hot potato. Did you ever play that as a kid where you pass the ball around and you try to get it out of your hands as quickly as possible because you don't want to be the one holding it when the buzzer goes off? And so that's essentially what's going to happen. It's going to be a game of hot potato. So NADH passes those 2 electrons to protein complex 1. Protein complex 1 accepts them, right? So it's become reduced. And then it's going to pass them on to this second molecule here. And so now complex 1 has lost them. So it's become oxidized. The second complex here has gained them. So it's become reduced. Then it's going to give the electrons to complex 3, who gives them to complex 4. And eventually, right, none of them want these electrons. It gives them to what we call the final electron acceptor, and that is going to be oxygen. This is why oxygen was required for this process, because it is the molecule that says, okay, guys, fine, I will accept these electrons. Okay, and so anytime a molecule accepts electrons, it has to do something with it, right? It's normally going to acquire hydrogen, protons as well. And so when oxygen, again, the final electron acceptor, when oxygen accepts those electrons, it actually becomes water. Okay, but oxygen, again, is what is actually accepting those electrons. Okay, so this was a series of redox reactions. Anytime one of these proteins gained the electrons, it was reduced. Once it passed them on to the next molecule, it became oxidized. Okay, the molecule receiving the electrons were gained. Once they lost them to the next molecule, it was oxidized. Okay, so it's just this series of redox reactions. So next, we need to think about FADH2, our other form of stored energy. Well, I think of FADH2, right? He was only made in the Krebs cycle. We don't get as much. I think of him as the second rate taxicab. So he's going to drop his 2 electrons off at complex 2. And it's going to be the same thing, another game of hot potato. They're going to pass these electrons from molecule to molecule until finally oxygen accepts them. Okay, so now we've handled our stored energy, all right? But hopefully you're sitting here going, hey, Chelsea, where's all this ATP you've been talking about? Well, let's talk about those. So in the membrane, let me find a color I haven't used yet. How about pink? What's going to happen is there's all these protons in the environment, right, naturally. Just draw a couple. All right, and so what's going to happen here is, remember back during our enzyme talk, we said that during any reaction that happens, a little bit of heat energy is released as a byproduct. And so with every redox reaction that's happening in this area, little bits of energy are being released in the process. So that energy is used to move some of these protons against their concentration gradients. This is a form of active transport, y'all. Okay, to move these protons against their concentration gradient. Trying to show that these guys have all been forced up here. Okay, so think back to our features of active transport, right? Where you're going from low amounts to high amounts. Okay, so make sure your picture kind of reflects that when you start. It's going against the concentration gradient anyways. Okay, so what's happening is all of these protons are constantly being forced across the membrane via active transport. So it's requiring energy, moving these protons against their gradient. But these little protons are sassy little boogers, okay? They do not want to be on the other side of the membrane. They want to be where they were originally. And so what they do is they all line up and they cross the membrane again using ATP synthase, okay? And it turns out that in the cell, ATP synthase, part of it is round and it actually works like a rotor. It physically spins in the cell. And so these hydrogen protons, as they pass through this enzyme, they're causing it to spin. And this is what is going to allow ADP to acquire that extra phosphate group to make ATP. 26 to 28 molecules of ATP. Now we're talking. Now we're mass producing some ATP. Okay, so again, as our electrons were being hot potatoed, right, across the electron transport chain, so through proteins 1 through 4, they were releasing little bits of energy, and that energy was being used to force some of the protons in the environment against their gradient. But because they're sassy, they didn't want to be there, and so they used ATP synthase, another transmembrane integral transport protein, to kind of get back to the other side. And in doing so, those protons, as they file through ATP synthase, they cause it to spin and it makes ATP. Okay, so now that you know what we're going to draw and what you want to have space for on your paper, I would go ahead and stop the video and kind of sketch out the electron transport chain, which again is the membrane, the 2 lines, and those 4 proteins, and this other one kind of here in the middle, you don't have to worry about its name. Okay, and then a little farther down, you're going to draw ATP synthase. ATP synthase is technically not part of the electron transport chain. Okay, the chemiosmosis that I mentioned earlier, that is the process of these protons going against their gradient and coming down ATP synthase to make ATP. That's chemiosmosis. I'm actually probably going to do another video where I have more of an animated slide to kind of help you visualize this process rather than a bunch of lines on the paper. But for now, just go ahead and do your best. So I'm going to erase a lot of my lines. So I will go through this again so you can follow along. I'm going to leave some of the details on there. All right, let me get rid of all these lines. Okay. Okay, so we're going to start with, let me get rid of the eraser so I don't erase everything I touch. Okay, so we have to do with our stored energy, which is NADH and FADH2. I think of them as competitors, and so they're not going to drop off their electrons at the same place. NADH2 is going to drop off its electrons at protein complex 1, and from there the electrons get hot potatoed or passed around down the electron transport chain until finally oxygen, the final electron acceptor, says, hey guys, I will accept these electrons, in which case it turns into water. FADH2 is doing the same thing. It's dropping off its electrons, but instead it's at complex 2. And so it is going to pass those electrons from one component of the electron transport chain to the next until again oxygen accepts it as the final electron acceptor. So that's the electron transport chain. It's literally a chain of proteins that are transporting electrons. And that's important because as these electrons are being transported, each redox reaction is releasing energy into the environment. And that energy is used to take these protons that exist in the environment and force them against their gradient, okay, through these transmembrane proteins here, okay, via active transport. But again, they don't want to be over there. And so what they do is they file, single file, down through ATP synthase, which allows that enzyme to mass produce ATP, getting about 26 to 28 molecules of ATP, bringing our net total to about 30 to 32 molecules of ATP, which is a lot. Okay. All right, y'all, that is, and I want to point out, this process is called oxidative phosphorylation. That's the way that we make the ATP. We're not doing substrate level phosphorylation anymore. We're using these redox reactions to phosphorylate ADP. So we say that ATP is made by oxidative phosphorylation in this step, hence the name of the step. All right, let me know if y'all have any questions.